Hello everyone, I'm Li Kunwang. I'm very glad to share you our recent study, Dietary Licorice enhances in vivo cadmium detoxification and modulates gut microbial metabolism in mice. Cadmium is a toxic non-essential metal element. In modern society, humans are exposed to excess cadmium in various pathways. Dietary interventions are the most practical approach for large-scale health interventions. In recent years, dietary interventions have been used for the prevention or treatment of multiple diseases, such as Alzheimer's disease and type 2 diabetes. Relative to modern pharmaceuticals, dietary herbs have low toxicity and side effects, and they are inexpensive and easily accessible. In this study, we searched and identified 5 dietary herbs: licorice, onion, fennel, ginger, and Sichuan pepper. The dietary herb containing food were made and supplied to the cadmium poisoned mice. After 1 month's intervention, we found that licorice led to a greatest decrease in the whole blood cadmium content among the 5 dietary herbs. We found that there was no significant change in food uptake in the dietary licorice treatments compared to the control. Licorice intervention reduced cadmium concentration in blood, heart, and lung, while it increased the cadmium concentration in liver and kidney. We suspect that licorice dietary enhanced the metabolism of cadmium in liver and kidney, resulting in the rapid removal of cadmium in other tissues and organs. We also found that licorice dietary relieved damage to hepatocytes caused by cadmium, improved liver function of cadmium-poisoned mice. The 2 main ingredients in licorice are glycyrrhizin and licorice. Licorice extract enhanced cadmium immobilization into hepatocytes. In addition, mRNA level of MT1 increased by dietary licorice in liver tissue. Metagenomic sequencing revealed that gut bacterial diversity was reduced by dietary licorice. A total of 18 key species were enriched in the dietary licorice group, especially Parabacteroides distasonis, which is a health beneficial gut microbe. Microbial metabolic function showed that the dietary licorice promoted the metabolic pathway of amino acids and bile acids, and increased the imbalance of glycoside hydrolase gene. Because glycoside hydrolase can transform glycyrrhizinic acid, the effect component for licorice, so we suspect that the bile transformation of intestinal microorganisms increases the efficacy of licorice. Licorice-derived metabolites, which showed on the right of the heat map, are positively correlated with the 18 key intestinal microorganisms. It is worth noting that L-type tyrosine, considered as a molecular marker of long-term cadmium exposure in previous studies, has negative correlation with most enriched bacteria. So from the figure we can see primary cholic acid such as TUDCA is transformed into secondary cholic acids such as DCA through genes such as BSH. Secondary cholic acid can regulate the synthesis of cholic acid in the liver, affect the composition of intestinal community and intestinal immunity, and regulate the fatty acid metabolism of the host. Therefore, we believe that licorice dietary intervention can promote the intestinal health of cadmium-poisoned mice. In summary, dietary licorice intake alters the diversity of mouse gut microbiome, increases the abundance of Parabacteroides distasonis, and enriches glycoside hydrolase genes and bile acid metabolic pathway. Glycyrrhizin is actively converted by gut microflora into inoxabolone. Inoxabolone is transferred into the liver and alleviates the liver damage caused by cadmium in terms of histopathological analysis and hepatic function. By sharply inducing MT1 gene expression, the liver and gut may have an interaction where bile acids, which affects the gut microbiome structure. As a result of cadmium accumulation in the liver, cadmium accumulation in other tissues was reduced. We want to thank all the authors who contributed to this publication and all our findings. Thank you for your attention.